My name is Christian Brender. I'm a cellular immunologist interested in antiviral T-cell immunity, particularly HIV, SARS-CoV-2, and herpes viruses. And we have been working with Omniscope on a number of projects involving T-cell receptor analysis in viral infections. And the one I would like to go through with you a little bit in detail is a study that we have conducted in an individual that we sampled prior to COVID infection, then after infection, after recovery from COVID, and prior to vaccination, then after vaccination, and even after a booster vaccination. So we had about 6 time points where we retrieved samples, did stimulate the cells with SARS-CoV-2 antigen to isolate really the virus-specific T cells, which we then subjected to OS-T, where we did T-cell receptor repertoire on 1 million cell per sample. And then we took this over time and started comparing repertoires. And this is shown here on this figure, that you see in dark colors, gray and blacks, the clonotypes, how they came up after a SARS-CoV-2 infection, then declined by the time the individual received vaccination, at which point the subject expanded a clonotype that was present upon infection, really as one of the major clonotypes in response to vaccination, which was then vaccine and waning a little bit over time and responding against to the booster vaccination. So this is just the big picture of how we can look at clonotypes. But what I would like to do is to really take you through a data set where we have identified these clonotypes in details. And so what you can do is you go into the immune portal of Omniscope, where your data are available. You can see here we have the data for the various time points, and we can start comparing repertoires across different time points. So if I were, for instance, to look at post-infection and post-vaccine, so pre or post-vaccination, essentially, we submit this and we get immediately the data back. This goes very quick. You can see that we had sampled at the various different time points, in each case more than 1 million cells, and we get between 650,000 and 1.2 million different clonotypes back, either as CD3 on amino acids or nucleotide-based analysis. We can then look into the length distribution, the abundance of this. The abundance is shown here. You see how often each clonotype is present. So this is in absolute counts here, where we have in per million clonotypes, some clonotypes that are present 2,000 or more times. But we can also look a little bit here more graphically, the clonal proportions. So you see that many of those clonotypes are present 1 or 2 times, and the minority is the very strongly expanded one in that set of 1 million TCR in a single sample. And we can compare this again between the time points that we have sampled them in. We can look at the CD3 length distribution as well. This is shown here, post-infection in nucleotides or amino acids, and we can compare that distribution then prior to vaccination with the distribution post-vaccination. What we can also look at in this Skanky plot here is all the clonotypes we want to select to further investigate. For instance, we have here the post-vaccine, a few clonotypes that are present in post-vaccination, and here a few that are present already after infection. And when we look, for instance, at this individual clonotype here, you see this is a clonotype that was present already in infection and then got further expanded by vaccination. We have other ones like this one here, which is a clonotype that is only induced upon vaccination, not surprisingly. that does exist, clonotypes that only are triggered by vaccination, or vice versa, that we have clonotypes that are only present after the infection. And that again may be explained by the fact that the vaccine does not drive all the clonotypes that the infection and the various proteins of the virus have triggered in the infection state, and that's not being picked up by vaccination. We can further visualize this a bit. These are now, each spot here is an individual clonotype. We can see the frequency here of the different clonotypes post-vaccination. On the y-axis, we have post-infection. And so anything that's down here on the bottom is clonotypes that were expanded upon vaccination, but they were not really present after infection, and vice versa. These are clonotypes that are uniquely present in the infection state, but were not carried over by the vaccination. Then we have anything in between that may have been carried over from the infection state and further expanded by vaccination. And this is the same showing here, just highlighting really the ones where we think these were present or not in infection and then expanded by vaccination. So this is really expanded by vaccination. We can readily zoom in here very conveniently. We can say we just want to look at what is highlighted here in orange. These are now all clonotypes that were expanded de novo on the bottom here by vaccination, or were present in infection and carried further into the vaccine response. And if we look, for instance, at a single individual clonotype like this one here, we can see that this clonotype is indeed in several databases. It has been related to surface glycoprotein epitope specificity. And so we can really just very simple and very easy click on each one of those as we are interested in to look at and identify whether it has been reported, whether it's a public TCR, whether it's more a private TCR, and what its antigen specificity is. And this, of course, goes way beyond COVID. As the databases are accessed with information from the sequencing here, we can identify their antigen specificity. So this was really a quick rundown on one example in one patient tested for SARS-CoV-2 specific T-cell responses over 6 time points involving an infection and the vaccination. And the information we can get from that really may help us to track vaccine-induced immunity over time in a very directly accessible manner, where we can look at individual clonotypes that are being induced by infection or not, or then picked up or not, or newly induced by vaccination. And so this will help us to accelerate vaccine development by knowing better what actually the vaccine candidate does in terms of immune repertoire and in terms of longevity of these responses and contraction of certain clonotypes or not. And that will help us to save time and money, because we do not necessarily need to run everything through extensive clinical trials. The application of this is, of course, goes well beyond T-cell receptor in infectious diseases. We could also use this to do a B-cell receptor repertoire, and that may be especially important for prophylactic vaccine development, where the B-cell immune receptor may be more relevant, as we see this possibly in HIV infection and prophylactic vaccines there. But it can also go beyond viral infections, for instance, in cancer vaccine, personalized cancer vaccines, or CAR-T approaches. So overall, I think the platform really sustains an easy, accessible way of looking at the T-cell receptor and B-cell receptor immune repertoires. I hope that was somewhat useful. Thanks for your attention, and I hope you get a glimpse of what OS-T and OS-B actually could do for your work.